Like everything in biology, the discoveries about protein synthesis were done in small steps, and it took us a while to actually figure out the chemistry of all of this. And well, as it was with many biochemical pathways, the way to discover how this actually works was by subsequently deleting enzymes which were part of the process, and then if you delete the enzyme, the process will stop at that point. And so you can know the order of when things happen and which parts are necessary for things to happen. And this takes many years to actually do. So, for example, you would get, let's say you start at point A, and then it goes to point B, and then it goes to point C, and then it goes to point D. If you delete the enzyme that's necessary to make the transfers from A to B, the whole process will stop at A, and you know that enzyme acted at that bridge there, and you know the order of which things will actually happen. And that's how we actually figured out the biochemical pathways, and much of the discoveries of protein synthesis were done in similar processes over many years by many different scientists. But early on, we didn't even know how can we possibly get this phenotype out of the DNA. But one thing seemed to be sure. We knew that DNA was somehow the key, but we knew that proteins were somehow defective out of DNA, and we didn't understand exactly how. What started for us people to understand how diseases were actually caused with a scientist called Archibald Gerald. And he looked at people who had alkaptonuria, which is an accumulation of chemicals in the body, caused several symptoms, including strange coloration in their eyes or in the skin, and also several other internal problems as well. And he figured out that all of that was because of the lack of an enzyme that actually broke down the chemical alkapton. And that means that he figured out that people who had this problem didn't really acquire this problem. It was more like they were born with some sort of inborn error in metabolism. And this thing that he called inborn error in metabolism will later on become known as mutations, which were later understood as a problem in the actual protein, either because a protein was badly made, or because transcription was badly done, or because the DNA itself had a problem. Another group of scientists later, called George Beadle and Boris Elfruzzi, worked together and experimented with Drosophila flies, the same flies that Thomas Hunt Morgan was working at, and they were actually interested in finding out why those flies had those different eye colors. And although they couldn't actually understand the exact biochemical pathway that was involved in the process, it was too early on for us to actually know all that chemistry. They knew one thing for sure. It seemed to be the lack of particular enzymes or problems in the protein that actually was causing the phenotypical difference in flies that had white color eyes. They lacked an enzyme that was necessary to complete the biochemical pathway. You see, for example, in the right side there, an example of a biochemical pathway and how complex it can be. Now, even though they didn't understand the whole pathway, they knew that the lack of certain enzymes was causing the problems in making the fruit fly have the white color eye. And that again confirmed the idea of Archibald Gerald of inborn errors in metabolism were leading to defects in the phenotype. Then, the same scientist, George Beadle, worked with another person named Edward Tatum and started working on basically a special type of mold to actually see what would happen if this mold was tried to be grown in different kinds of environment. They came up with the idea that if you gave the mold the minimal environment, that means only the things that the mold actually needs to survive, most mold can actually deal with that just fine, and they will survive with no problem at all because they have normal genotypes. But if you mutate some of the molds, maybe by blasting them with radiation, then they will gather mutations and be incapable of processing the chemicals they need to process out of that minimal medium. And they will only be able to grow on a complete medium, which has everything they possibly could use, so that they don't have to actually create the stuff they need from scratch out of the minimal medium. And so, although the mutants were able to survive on a complete medium, they failed to survive on the minimal medium because they lacked certain enzymes that could make the job possible and make the products that they needed out of the minimal medium. Another group of scientists called Adrian Spur and Norman Horowitz actually got together to actually look at the process that Edward Tatum and George Beadle had worked on to try to identify the actual biochemical pathway that was defective, causing the mutants to be unable to produce the arginine that they needed from the minimal medium. And they actually identified there was actually different classes of mutants. and that some mutants were lacking the very first enzyme on the step of the mechanism. Others were lacking the second enzyme. Others were lacking the third enzyme. And then by doing this, they figured out the actual order of the biochemical pathway that led from the formation of arginine from the minimal medium. Some mutants were able to get all the way to the second step because they were missing the third enzyme and they couldn't do the last step to make arginine. Other mutants were missing the stop at the first step because they couldn't even do the first step because they were missing the very first enzyme. And then they came up with this idea that genes that were inside the spore were being mutated by the X-ray blast, and with that they were messing up the production of the enzymes that were necessary later on to make the successive growth of the spore in the medium. And so it came in the idea of the one gene, one enzyme theory. Since then, however, we have changed that theory because we now understand that the genes code not just for enzymes, but for multiple things. Proteins have multiple jobs, including regulation, immune system, structure, movement, not just enzymic or catalytic roles. Therefore, we changed it to the idea of one gene, one protein, or one gene, one polypeptide kind of idea. But we have also changed that again nowadays because we now understand that sometimes proteins require multiple genes to make it, since they are assembled out of different subunits or different domains. Remember, we talked about that in the beginning of the lecture series. So that's where we are at right now. The idea that you can get to a protein, you need one or many genes, and that sometimes one gene can become multiple proteins because of differential RNA processing. Remember that? So that's how we got from the understanding that DNA can make proteins, because we understood that when we force the mutation on the DNA by blasting the DNA with X-rays, you also cause a problem in the protein that's made. In other words, changing the DNA changed the protein, and that's how we got to the idea that proteins are based on DNA. Now, how do we actually figure out the process of gene expression or protein synthesis? Now, like I said, it was based on a sequence of biochemical reactions where they looked at enzyme after enzyme to see where the process would stop if you took that chemical away. But without getting too much detail on that, because that would take forever, because think about how many steps we talked about, let's just talk about one crucial step that was made in the 60s, actually, by a scientist called Marshall Nirenberg, and he ended up winning a Nobel Prize for this. He figured out the actual genetic code. Remember that codon table we learned about? But how did he do that? The way he did this is that he got several transfer RNAs and put them in a big soup. And he also got a bunch of amino acids and put it there. He also put the ribosomes. He put everything that was necessary for protein synthesis to take place. But he didn't put any messenger RNA. He only put a small snippet of messenger RNA, or literally one at a time. So only one codon at a time. And he started this with UUU. And he wanted to see, I wonder what UUU stands for. What does this message say? What does this codon mean? Well, the first thing he did is to figure this out is that he used what is called a radioactive filter. This radioactive filter would only block the transfer RNA that was connected to the anticodon of interest and surrounded by the messenger RNA and the ribosome. So in other words, all the other transfer RNAs would not be caught by the filter. But if the transfer RNA attaches itself to the anticodon of interest and the ribosome form itself around that, then you're actually going to get that be caught up on the radioactive filter. Then he also went on a step further and he radioactively tagged the amino acids that he put in the medium, one at a time. So he put one codon at a time and tagged one amino acid at a time. So let's say, for example, example A, when he tagged PRO amino acid and he put UUU at the same time. Since UUU is the codon that stands for the anticodon AAA, only the amino acid that carries PHE would actually be tagged on the radioactive filter. So he didn't see any results when he did it that way. But when he changed it and started tagging different amino acids, he tried it again and again until finally he tagged PHE. Once he tagged PHE, he actually noticed that PHE would stay at the radioactivity filter. And then what he figured out then is that UUU paired up with AAA, which carries PHE. And so he understood that UUU is the codon for the anticodon AAA, which actually is on the transfer RNA, which specifically carries PHE. And that's how he figured out that the RNA code UUU means PHE. And he repeated this process over and over again for different codons, every one of the 64 codons in the sequence, tagging every single one of the amino acids until he found out the actual pairing between the codons and the amino acids. And that took him a long time, but it won him a Nobel Prize. And that's the science that was done to figure out the genetic code that seems to be pretty much universal. And then after that, we did successive trials and they figured out the whole protein synthesis mechanism, which involved the messenger RNA, the transfer RNA, and the ribosomes, all by deleting molecules which are necessary every step of the way and seeing where the process would stop in the chemistry of the cell using tags and everything else that we talked about. So that's protein synthesis in a nutshell. I hope you learn a lot and don't do anything that would not make your mama proud.